Good morning, everybody. Good morning. How you guys doing? Having a good visit?
Yes.
No frowns. No frowns are allowed here. Everybody has to smile. It's the price of admission. So my name is Kevin Ahern, and I am a person at OSU who wears a couple of hats, which for my bald head are kind of important because out in the sun you sunburn pretty easily. I am a professor in biochemistry, biophysics at Oregon State, and I'm the head advisor for students in that major. And so I'm very passionate about biochemistry. Today I'm going to give you your first college lecture, and it's going to be about biochemistry. And I promise to keep it not too technical. And I like to design these so that there's something that everybody can understand something about biochemistry. So we'll do that. But also I can assure you we'll have a little fun too, because I like to have a little fun when I lecture. I think there's nothing worse than a boring college lecture. And if your first college lecture is very boring, that's very bad. And boy, I'm getting a lot of feedback there. Can you turn this off? Can you turn that guy off? Yeah. Okay. The other hat I wear is I'm a director for undergraduate research at OSU. So my job is to get students involved in undergraduate research. And I like to get students out there. What students find surprising is you can be in any major and do undergraduate research. You can be an English major, you can be a history major, you can be a biochemistry major, you can be an engineer, you can do undergraduate research. And you can start that as early as your freshman year. So if you're interested in that sort of thing, please talk to me afterwards and I'll be happy to tell you more. Okay, well, as I said, this is your first college lecture, and I've already pushed the wrong button. I've already failed my first college lecture. I promise I won't give you a pop quiz at the end, but I also promise to get you involved. Okay, so you're not going to be able to just sit around. I think we talk about active learning. We talk about active participation. So I have a way to actively get you to participate in your learning process. We'll do that today. Well, the lecture I have for you today is they call it's in your blood. And let me introduce this topic by saying, well, people say, well, what do biochemists do? What do biophysicists do? I'm a biochemist, biophysicist. What do they do? Well, if we think about biologists are interested in the whole organism. They look at organisms. That's the topic of their study. Physiologists, by contrast, are interested in tissues and what's happening within that organism. Microbiologists, by contrast, are interested in cells. Okay? So they get smaller and smaller and smaller. So we've started with an organism, a human being. We've gone down to the size of a lung, and then down to the size of a cell. And biochemists go smaller than that. Biochemists and biophysicists are interested in the molecular basis of life. Okay? So we get down to the molecule level. All right? And I'm not only going to take you down to the molecule level, but I'm going to take you down smaller than the molecule level. Okay? So we're going to get really down and dirty with this. Hemoglobin is the subject of what I'll be talking about today. And you probably think you know everything there is to know about hemoglobin. But what I hope to do is to surprise you. And I hope to teach you something about the amazing protein that's known as hemoglobin. Okay? So that's the topic of my talk today. Hemoglobin is a protein. It's not only one protein, it's actually four proteins. It has four subunits. It has a subunit here in red, another subunit in red, a subunit in blue, and a subunit in blue. The two blues are identical. The two reds are identical. The two reds are called alpha subunits, and the two blues are called beta subunits. Okay? You don't need to know that. As I said, there's not a pop quiz today, right? So, what you should recognize is there's something else that's on there. That's these little green things. And these little green things are the things that give hemoglobin its name. They're known as a heme. Okay? So heme is a very interesting structure. And if we look at heme up close and personal, we see something that looks like this. Okay? This heme is actually a sort of a flat ring-like structure. Okay? Heme is the most important part of hemoglobin, as we will argue. But what we see within heme is that in the very center of it, there's an atom of iron. Okay? Now this atom of iron is one of the reasons we have to take iron and have to get iron in our diet. This iron plays a very critical role in hemoglobin because what this iron does is it's the guy that grabs onto the oxygen and hangs on to the oxygen for you. So as the hemoglobin is passing through your lungs, okay, the iron grabs that oxygen and hangs on to it. Later, when the hemoglobin leaves the lungs and goes out to the tissues, the iron lets go of the oxygen, and the oxygen goes to your tissues that need it. Okay? This is a really cool system. I'm going to show you some things about it that's probably pretty amazing, I think, and probably going to amaze you as well. All right. Now, as I look at this, here's the iron right here in the middle where this little star is, and we can see that the iron is actually attached to something down there. Okay? So you don't need to know this, but proteins are made of amino acids. You've heard of amino acids. I'm sure everybody's heard of amino acids. Well, these guys are all amino acids, and this is one of the amino acids right here. And you can see that this guy right here is attached to the iron, and it's attached to the rest of the protein. Keep that in mind. Okay? So ultimately that iron is attached to the entire rest of the protein. Okay? That's cool. All right. Well, here is looking at that flat ring structure from the side. Instead of looking down on it where you see the top of it, you're looking at it from the side, something like this. Okay? You see on the left side that we have something called deoxyhemoglobin, and this is what that ring structure looks like in the absence of iron. When there's no iron, that's what it looks like. And when it has when it has no oxygen, that's what it looks like. And when it has oxygen, we look at it on the other side, and we see there's actually been a slight change. Now that change is extraordinarily slight. Extraordinarily slight, okay? You notice that there's this little thing over here that says 0.4 angstroms. I won't tell you how small an angstrom is, but trust me, an angstrom is enormously small, if that makes any sense, right? Talk about your oxymorons, right? Okay, it's enormously small. All right, it's an incredibly small thing. What's happening in this process is when oxygen comes into the hemoglobin protein, okay? Oxygen binds and iron moves incredibly tiny distance. An incredibly tiny distance. Even with there's no way to see that. Okay, we can't visually see that. Okay, we have to measure it. But this incredibly tiny distance is one of the most important things in your entire body. This change that I just described to you enables you not only to be alive, but enables the life of animals on the face of the earth. Okay, without this change, that will not happen. Now I'm going to tell you why, okay? But without that change, that does not happen. Well, why is that important? Well, let's think about this. You notice I showed you 4 subunits. I showed you 4 rings. I'm showing you 1 ring. What happens in 1 ring is going to influence the other 3. Okay? Well, what happened? The iron moved a very tiny distance up. It got lifted up. So instead of being something that looked like this, it flattened very slightly. The iron moved up a very tiny distance. Well, the iron is not unattached. The iron, you recall, is attached to the entire rest of the protein. We have 4 proteins in hemoglobin. We're looking at 1 of them. Okay? Well, the toe bone's connected to the foot bone, and the foot bone's connected to the ankle bone, and the ankle bone's connected to the shin bone. If I pull on the toe, what happens? I change everything. I can't pull on one thing and not change everything else. Right? So I've just changed very, very tiny amounts the structure of that 1 protein of the 4 inside of hemoglobin. Okay? Well, that 1 protein inside of hemoglobin is interacting with the other 3. And as a consequence of that 1 protein being changed, the other 3 change. Okay? So we're seeing literally a chain reaction that's going on. We're seeing something that's affected 1 of them, and now the other 3 are affected. But what's the effect on the other 3? Well, to tell you that, I need to tell you something about how hemoglobin works. Hemoglobin, as it passes through your body, is designed to deliver oxygen. So we're going to start out in the lungs. Hemoglobin gets to the lungs, and hemoglobin is in the lungs for a very brief time. Your blood flows pretty fast. It's flowing pretty fast. That hemoglobin has got to pick up oxygen when it's in the lungs in that fast time that it's there. When hemoglobin gets to the lungs, it doesn't want to bind oxygen. That may seem a little surprising to you, but it doesn't. It's in something we call the T state. Okay? The reason it binds 1 oxygen is the oxygen concentration in the lungs is so high that it literally forces itself onto 1 of those subunits. And 1 of the subunits begrudgingly takes that oxygen, like I just described to you here, and the process becomes changed. I've told you that change now caused the other 3 subunits to change, and now I'm going to tell you how they change. They change from not wanting to have oxygen to wanting to have oxygen very bad. Okay? Now this is really critical, because remember, the blood's moving very fast. We've got to get that oxygen loaded up in hemoglobin while it's passing through. So it's really nice if we get hemoglobin into that state where it wants to bind oxygen so it can grab it while it's there and goes out to the rest of the tissues. I said this was important if you're an animal, because animals have a blood flow so they can deliver oxygen to tissues that need it. Animals have muscles. Animals use muscles to catch things. Animals use muscles to run away from things. Animals use muscles to do all kinds of things. All right? Muscles need oxygen. Okay? Plants don't need oxygen. Plants can get the oxygen. Plants make oxygen when they do photosynthesis. Plants have all the oxygen that they need sitting around. They don't need a blood supply to do that. Okay? We've got to be able to load up our hemoglobin very quickly and very efficiently for us to be able to feed our muscles. Okay? So, we've fed our muscles now as a result of this very tiny change that's happened right here. Okay? Pretty cool stuff. Well, I'm a scientist. I can't show you a lecture without showing you a graph or two. Okay? So I'm going to show you a couple of graphs. All right. What's this telling us? Well, what this is showing how oxygen in the body changes relative to its being bound to hemoglobin. All right? What the y-axis here is showing me is just telling me how much of the hemoglobin is full of oxygen. That's 100%. That's 0%. Okay? And down here, I see the concentration of oxygen. So I see as the concentration of oxygen increases, as might happen in the lungs, the amount of hemoglobin bound to oxygen increases up to 100%. That's very important. That's very cool. And it tells us something that we kind of say, well, of course I know it binds oxygen, right? So what's the big deal? All right? Well, the big deal is that hemoglobin not only has the ability to grab a hold of oxygen, but as we will see, it has the ability to let go of it. You've got another protein in your body called myoglobin, and myoglobin has a great ability to grab a hold of oxygen, but it doesn't have such a great ability to let go of it. Your body uses it like what I call a battery. Okay? Your laptop has a battery in it. You unplug it from the wall, the laptop continues to function because the battery kicks in. Myoglobin is related to hemoglobin. But instead of being able to let go of oxygen very well, it's very good at grabbing it and holding on to it. The only time it lets go is when the oxygen concentration gets very, very, very low. Our muscles use oxygen as that battery. When our muscles start running out very, very, very low of oxygen, myoglobin starts to give it up. But most of the time it doesn't. It holds on to it like your battery holds on to juice in your body, in your laptop. Okay? Well, myoglobin doesn't do a very good job of having two different states. One state where it's loaded with oxygen and one state where it's letting go. Look at hemoglobin. Okay? If we compare the same oxygen concentration, here hemoglobin's got about 50% and myoglobin's still up close to 100%. This is telling us that is when the hemoglobin is traveling through the body to a place where there's not much oxygen, what's it going to do? It's going to let go of oxygen. That's really good. So as the oxygen concentration starts to fall, hemoglobin's going to give it up. Well, that's really important because we have a variety of levels of oxygen in our body. And hemoglobin is very well set up to deliver oxygen at places where it's needed and to pick it up at places where oxygen's abundant, as we have in our lungs. Okay? Really, really important stuff. This shows the two states of hemoglobin that I've referred to just previously. Hemoglobin is present in two states. One state is called the T state, I've already referred to. The T state is a state where hemoglobin really doesn't want to bind oxygen. If it's bound to oxygen and it flips into the T state, what it will do is let go of it. Okay? So if it flips into the T state, it will let go of it. When it gets into the R state, it loves to grab a hold of it. So what's happened with what I described in the lungs is that one subunit grabs a hold of oxygen, it changes the other three, and the other three want to bind it more. That phenomenon we call cooperativity, meaning that one binding of one is affecting the binding of the other three. That's very cool. Okay? And that flipping is what's happening. What's happening is they're flipping from the T state into the R state, and in the R state, they want to grab oxygen. We call that high affinity. Okay? So we want hemoglobin to have high affinity when it's in the lungs, and we want it to have less affinity as it's passing through the body. Okay? Now, hemoglobin actually has structural differences. You can actually see differences in the way these two guys appear. All right? that T and R state has a physical basis. They're different fundamentally in terms of structure. There's something else I want to show you here. I'm going to exaggerate it for you in a slide in just a second. And that is, if we look at hemoglobin like we see here, all right, we see that in the middle it's kind of hollow. I like to describe it as sort of a donut. Okay? Sort of a donut that's there. All right? That donut turns out to be really critical for what hemoglobin does in the body. Okay? How many people in here have ever heard of a molecule called 2,3-BPG? I never get an audience that says yes. Okay? This is one of the most important molecules in your body. It's one of the most... That's fine. It's not a pop quiz. And this is why I say it's a good reason to take biochemistry. Okay? You'll learn something. I'm going to teach you something today about this molecule that's going to leave you, whoa, that's really interesting. If not, I didn't do my job very well. Okay? So, 2,3-BPG. What is it? Well, chemically, that's what it looks like. There's the full name, blah, blah, blah. You don't care about that. I don't care about that. Okay? The most important thing about this molecule is what it does. Okay? This molecule is something that our body produces as a byproduct of metabolism. Metabolism is the business of being a cell. Okay? Something that has a high metabolism might be a muscle cell that's exercising. The more it exercises, the higher its metabolism, and the more of this molecule it produces as a result. We can think of this molecule as being a sort of flag that tells the body, hey, here is where rapid metabolism is going on. Okay? So my muscles, as I'm flexing and bending, or using my muscles, are producing 2,3-BPG. My skin, which is sitting here doing nothing but laying around and maybe catching a ray or two, is not producing much 2,3-BPG. Okay? A lot of 2,3-BPG in my muscles. Why do I tell you that? Well, 2,3-BPG fits beautifully into the donut hole of hemoglobin. Okay? It fits beautifully. This just shows you technically how it happens, but all we really care about here is it fits really nicely into that donut hole. When 2,3-BPG binds to hemoglobin, it causes it to change. Now let's think about what's happened to hemoglobin. Hemoglobin went through the lungs. It's full of oxygen. So what state's it going to be in? R or T? It'd be in the R state, right? Because it was full of oxygen. It had great affinity for it. It's hanging on to that oxygen. Okay. It hits the tissues where the 2,3-BPG is being produced. We really would like to have hemoglobin let go of the oxygen. And so what has to happen for hemoglobin, or one of the things that can happen for hemoglobin to let go of the oxygen? It's going to flip into the T state, right? Well, it doesn't flip all by itself. Guess what causes it to flip? 2,3-BPG. Now this is really very nice. It's set up very cool because what happens is the place where the oxygen is the most needed, the cells that are metabolizing the most rapidly, are sending out a signal, and hemoglobin is recognizing that signal, and it's saying, oh, you need some oxygen here, I'm going to let you have some oxygen. Pretty cool stuff, right? All right. This shows schematically what happens if we have hemoglobin without 2,3-BPG and hemoglobin with 2,3-BPG. Okay? If we look at the concentration of oxygen in the tissues, look at the difference. Okay? With no 2,3-BPG, 8% of it has given up its oxygen. In the presence of 2,3-BPG, 66% has given up its oxygen. Meaning that 2,3-BPG is telling hemoglobin, let go of your oxygen, and it's converting it into the T state. Cool, right? Well, maybe not right. Okay? We just converted hemoglobin into the T state, okay? And we've got, not only have it in the T state, but we have something locking it in the T state. 2,3-BPG. If that 2,3-BPG gets stuck in the hemoglobin and it makes it all the way back to the lungs, and there's still 2,3-BPG there, what's going to happen? It can't flip into the R because 2,3-BPG is holding it in the T state. Well, fortunately that doesn't happen. 2,3-BPG does not bind to hemoglobin irreversibly, meaning that it can come off and so forth. As it comes off, it sort of does an exchange. It comes off, it goes on, it comes off, it goes on, it comes off, it goes on. And every now and then when it comes off, a cell grabs it. So as it's traveling on its route back to the, its hemoglobin is traveling on its route back to the lungs, 2,3-BPG at various places is literally being pulled away by cells. Okay? Now that's really cool, and that's really important, because as that hemoglobin gets back to the lungs, it doesn't have 2,3-BPG, it's still in the T state, but it's not locked in the T state. Getting technical here, aren't I? I promised I wouldn't, and here I am. Okay. All right. It's not locked in the T state, but it's not stuck with 2,3-BPG. It gets to the lungs, that first oxygen binds, cooperativity, and the whole process goes on again. Really cool, really beautiful, except one thing, and that is if you're a smoker. Ooh, okay? Smokers, in their bloodstream, have a much higher level of 2,3-BPG than non-smokers do. Now, I think you can probably put the pieces together and figure out what happens with this. The smoker is much more likely to have their hemoglobin make it all the way back to the lungs, loaded with 2,3-BPG, and when it's loaded with 2,3-BPG, it can't flip out of the T state, and consequently, it doesn't bind much oxygen. 2,3-BPG is why smokers get out of breath when they climb stairs. Okay? That's the reason, or one of the reasons, why they get out of breath when they climb stairs. There's another reason why smokers get out of breath when they climb stairs. Okay? I didn't tell you, but I showed you oxygen binding to the iron in the hemoglobin, at the heme, right? You saw that iron, you saw that those two little atoms up there of oxygen binding to it? Well, it turns out that there's another molecule that looks almost identical to oxygen that'll bind there as well. Carbon monoxide. Smokers get a lot of carbon monoxide in smoking cigarettes. Okay? Two reasons not to smoke. What smokers are literally doing is suffocating themselves from oxygen. They're starving themselves of oxygen in that process. Okay? Smoking has other dangerous dangerous side effects from cancer and so forth. But from an oxygen perspective, smokers are killing themselves because they're not getting enough oxygen that they need. Okay. Question? Yeah.
So, on the other side, like a long distance runner
Yep.
who's trained extensively, how is their
Good question.
mechanism changed?
Good question. So long distance runner, they've trained extensively, and they probably have a little different aerobic capacity than a person who hasn't trained extensively. We see this happen in a couple ways. One is with runners. Another is people who live at high altitudes. People who live at high altitudes have a lower oxygen concentration, and they need to deliver oxygen. They have more ways to grab oxygen. Both will have increased levels of hemoglobin. So I'm a jogger, and I like to run with my students. And I had a student. He went and he studied in Ecuador for about 6 months, and he was in keto, way up in the Andes, 10,000 feet or so. And when he was running with me, I had to struggle to keep up with him, but I managed to keep up with him. When he came back, it was incredible because his body had adapted in that 6 months where he was making more hemoglobin, and he said he did anything, he didn't get out of breath. So that's the adaptation, one of the adaptations that happens in that process. Yeah?
Erythropoiesis.
Yes, erythropoiesis. That's the making of red blood cells. You betcha. You passed the pop quiz. Okay, I'll let you out without the pop quiz today. Okay, good. Other questions? All right, well, if that were all there was to hemoglobin, it would be a pretty amazing protein. But there's more. Okay? But wait, there's more. All right? And the more is something called the Bohr effect. The Bohr effect is also showing us how hemoglobin is amazingly tuned to the body's needs. One of the molecules I said that was a flag that says, hey, things are needed here, was 2,3-BPG. You saw in that graph I showed you that at the tissue level, we went from 8% down to 66% of the oxygen delivered. That meant that 1/3 of the hemoglobins still had oxygen, even at relatively low concentrations of oxygen. We'd like to dump all that. We want to get that out of there. Okay? Well, the Bohr effect helps that in addition to happen. Okay? This shows what's happening. This shows what's happening with a metabolizing tissue, dumping off the byproduct of metabolism. The byproduct of metabolism is carbon dioxide. Okay? Carbon dioxide turns out to be pretty poisonous. Okay? We got to get it out of our system. Your carbon dioxide levels get too high, and you will die, and you will not die a pleasant death. Okay? So it's important to get rid of the carbon dioxide. The Bohr effect explains how the body gets rid of carbon dioxide and how hemoglobin participates in that process. Carbon dioxide can readily, in the bloodstream, be converted into something called carbonic acid. There's an enzyme that does this. You hear about an enzyme catalyzing a reaction, right? Everybody's heard about that. An enzyme catalyzes a chemical reaction. The chemical reaction that it catalyzes is this guy right here. Carbonic anhydrase, the enzyme that catalyzes this is amazing. It's called carbonic anhydrase. Okay? Carbonic anhydrase, if I take one single enzyme, not 50, not 100, not 1,000, but one single carbonic anhydrase, and I mix it with some carbon dioxide and water, in 1 second, one enzyme will convert 1,000,000 of these. There's no way on earth I can conceive of doing anything 1,000,000 times a second. It tells us that at the nanoscopic level, life and nature is very different than what we see in the macroscopic world. Okay? Enzymes are remarkable, but I'm not here to talk about enzymes. What this guy's doing is it's converting and it's making some acid. Protons, wherever you see that, that's a proton. Okay? Protons are acid. You've heard about acid stomach and all the various things. Acid is another indicator of metabolism. Rapidly metabolizing cells are making carbon dioxide. Rapidly metabolizing cells are making acid. Okay? Well, what happens as a consequence is it turns out that if we make more acid, we cause hemoglobin to let go of more oxygen. Here is how much it holds at a pH of 7.4. There's that 66%. And here's how much it holds at a pH of 7.2. It has dumped another 11% of oxygen. Okay? The body is picking up those signals. Those protons are telling hemoglobin something, and hemoglobin's giving up that oxygen at the places where it's needed. Not only that, carbon dioxide will do the same thing. Here is if we compare the pH 7.4 with pH 7.2, and now pH 7.4 and 7.2, and we add some carbon dioxide, and look what happens. We lose even more. Okay? So the things that are byproducts of metabolism are signaling to hemoglobin, here's where metabolism is occurring, let go of your oxygen here, and the body's needs are being met as a consequence of that. Okay? The Bohr Effect explains all that in an amazing way. Okay. This shows it in a very technical capacity. I'm not going to show it to you in a technical capacity, but it does show something over here that is important. Hemoglobin carries the carbon dioxide. It actually physically picks it up and carries it back to the lungs. And in the lungs, the reversal of this process happens, and carbon dioxide is exhaled. If this didn't happen, again, we're dead, because we die of carbon dioxide. We acidify and we die. Okay? We've got to get the carbon dioxide out. Hemoglobin is participating in that process. So hemoglobin is carrying oxygen from the lungs to the tissues. It's carrying carbon dioxide from the tissues back to the lungs. Amazing protein. Absolutely incredible. But that's not all. Okay? Hemoglobin is just exploding with things. Okay? Think about the fetus. Mom is pregnant. Okay? She is the source of nutrients for the fetus. She is also the source of oxygen for the fetus, which needs oxygen to grow. If the fetus has the same hemoglobin as mom does, the fetus is fighting a losing battle. Okay? If they're the same, it's hard for the fetus to take oxygen away from mom. It turns out when the fetus is in mom, has a different kind of hemoglobin. It's known as fetal hemoglobin. It's very slightly different. I told you that the adult hemoglobin had alpha-2, beta-2. The fetus has something called alpha-2, gamma-2. The betas are substituted for gammas. The gammas are very, very much like the betas, but they're not identical. The difference is that the fetal hemoglobin has a greater affinity for oxygen than mom's hemoglobin. Meaning the fetus can literally take oxygen away from mom's hemoglobin and do its thing. Now the way that it does that is kind of interesting. It turns out that fetal hemoglobin, you remember that donut in the hemoglobin? The donut in the fetal hemoglobin is smaller. 2,3-BPG won't fit in it. 2,3-BPG can't force it to go into the T state Fetal hemoglobin stays in the R state, and what does the R state tell us? Greater affinity for oxygen. That's how the fetus is taking oxygen away from mom. Pretty cool stuff. Sickle cell anemia. Everybody's heard of sickle cell anemia. Okay? Sickle cell anemia is a genetic disease. It arises as a result of a variety of different mutations, but it is a debilitating disease. What happens in sickle cell anemia is a person who has this disease has a hemoglobin that will tend to cause blood cells to sickle, form a sickle shape under low oxygen concentrations. That is the place where once it's delivered its oxygen into the tissues, it can turn from this nice smooth shape into this very nasty looking shape. This very nasty looking shape right here gets stuck in the capillaries. This guy doesn't. One of the reasons we have nice rounded blood cells is so it can flow through our capillaries. They're flexible. They don't get stuck. This guy gets stuck. People who have sickle cell anemia, if they do heavy exertion, find it can be very painful because their muscles are completely starving for oxygen. They're not getting oxygen delivered to them, and more, their capillaries are getting plugged up by these sickle cells that are getting stuck within them. Okay? Really debilitating disease. The reason the sickle cells form is hemoglobin, under those low oxygen concentrations, actually forms polymers. Long threads of hemoglobin. What you see in this electron micrograph are some of those threads of hemoglobin, and it's these threads that cause that sickling to occur. Hemoglobin doesn't do that in a person who doesn't have sickle cell anemia. Well, this is very interesting, and people wonder, well, this is a very debilitating disease. How come it's perpetuated in the human population? And when they compared the highest incidences of sickle cell anemia with the prevalence of malaria across the world, they found the two maps overlap pretty amazingly. And so that got them wondering, was it possible that there was an advantage to sickle cell anemia for people who had malaria? And what they discovered was, in fact, there was. People who are heterozygous, meaning they have one good gene and one bad gene for sickle cell anemia, have a protective effect against sickle cell anemia. They actually, especially when they're younger, are much more likely to survive malaria if they have that than if they don't have that. So, something we look at as a genetic disease, something that obviously causes problems, is there because it has an advantage. It conveys an advantage to the people who have it. Okay. People like this one. Okay. There are diseases, okay? There was a disease that it's actually fairly well known, where people produce too much heme, okay? And as a consequence, they have some unusual properties, okay? One of which is their skin turns a different color. It's sort of a bluish color. They tend to have difficulty with being out in bright lights, okay? Because the light and the things that they absorb cause problems for them. So that's a real problem. And it is thought that perhaps the legend of the vampires being afraid of going out in the light and so forth arose from the actual physical condition that people who had this disease, where they were producing too much of this. Okay, this disease causes people at times to go literally insane. They will lose their mind for a period of time. King George III of England was thought to have this disease. And it's thought, again, thinking back to the vampires, how they suddenly are taken over by something. Perhaps this had its roots in the disease relating to hemoglobin. Kind of cool stuff. All right. Well, one of the things that I like to do, as I said, is engage you. And that's the last part of what I have to say today. I'm going to engage you, and we're going to do something that I like to do with all my classes, which is I like to drive home the message to you. Okay? So you're going to help me with this. These are songs I write about biochemistry. They're called Metabolic Melodies, and we're going to sing them. The first is called Hemoglobin's Moving Around. We're going to learn the lesson from today. Oh, isn't it great? Proteins can do, especially ones that bind to O2. Hemoglobin's moving around. Inside of the lungs, it picks up the bait and changes itself from T to R state. Hemoglobin's moving around. The protoporphyrin system Its iron makes such a scene, arising when an O2 by pulling up on histidine. The binding occurs cooperatively thanks to changes quaternary. Hemoglobin's moving around. It exits the lungs, engorged with O2, in search of a working body tissue. Hemoglobin's moving around. The proton concentration has high and has a role between the alpha beta. It finds a midazole. The Lord. Oh no! I screwed that up. Sorry. Keep going. Now I totally screwed it up. Let me start over. Once it starts, it won't finish. I failed my pop quiz. Ones that bind to O2, hemoglobin's moving around. Inside of the lungs, it picks up the bait and changes itself from T to R state. Hemoglobin's moving around. The protoporphyrin system, its iron makes such a scene, arising when an O2 binds, pulling up on histidine. The binding occurs cooperatively thanks to changes quaternary. Hemoglobin's moving around. It exits the lungs, engorged with O2, in search of a working body tissue. Hemoglobin's moving around. The proton concentration is high and has a role between the alpha betas. It finds a midazole. Wait for that. Empty their loads, the globins decree. We need to bind to 3-BPG. Hemoglobin's moving around. The stage is thus set for grabbing a few cellular dumps of CO2. Hemoglobin's moving around. And then inside the lungs it discovers oxygen and dumps the CO2 off. Start all over again. So see how this works? You better expect to have to describe the Bohr Effect. Hemoglobin's moving around. You get into that. That's not me. All right, but wait, there's more. Okay. I'd like to reinforce things, okay? Anybody know the old song for the older people here? Like, Jen's inside my porphyrin rings, nudge the irons up a notch and yank on histidines. The globin shapes will change a bit. Oh, what a sight to see! The way they bind to oxygen cooperatively. And as I exit from the lungs to swim in the bloodstream, metabolizing cells, they all express their needs to me. To give up oxygen and change from R to T. Well, my amines, they hang on to the protons readily. That's not all the tricks I know. There's more that's up my sleeve. Perhaps between subunits that hold 2,3-BPG. when near metabolizing cells I bind things that diffuse the protons and bicarbonates from lonely CO2s is when your cells are at play. Go say hip hip hooray for the bloody things. That's the way it is. Okay. All right. The last one you're going to help me with. All right. I need your help. I need some clapping. Ready? Not too fast. Not too fast, or I'll get in trouble. You're going to sing with me. Everybody know "Thank God I'm a Country Boy"? All right. Ready, set, go. There's a bundle of things a student ought to know, and Ahern's talk isn't really very slow. Learning ain't easy, the lectures kind of blow. Thank God there's a video. Well, we've gone through the cycles and their enzymes too. Studying the regulation, everything is new. I gotta admit that I haven't got a clue. What am I gonna do? So I got me a note card, bought me a strier, got the enzymes down and the names he requires. Hope I can muster up a little more desire. Thank God there's a video. Just got up to speed about the NAD. Protons moving through complex V. Electrons dance in the cytochrome C. Gotta hear the MP3. Fatty acid oxidation makes the acetyl-CoA inside the inner matrix of the mitochondria. Guess it's very complicated, I guess I gotta say. Thank God there's a video. So I got me a note card, bought me a strier, got the enzymes down and the names he requires. Hope I can muster up a little more desire. Thank God there's a video. Replication's kind of easy in a simple kind of way. Copying the bases in the plasma DNAs. G's go with C's and T's go with A's. Thank to polymerase. And the DNA's a template for the RNA. Helices are winding at TA. Termination happens and the enzyme goes away. Don't forget the poly A. So I got me a note card, bought me a strier, got the enzymes down, the names he requires. Think I can muster up a little more desire. Thank God there's a video. Okay. Thank you. If you're interested in the metabolic melodies, these are songs I sing for my students. I've got over 150 of them. That's the URL. And they're all free. All the songs are free. I give them away. And for anybody, I have CDs of the songs. I will give away for free to the first 6 people who come up here. Thank you.